Cells are the building blocks of life, and we need them to divide in order for us to grow and to repair. Our bodies contain trillions of cells and must all divide accurately to pass down the correct genetic information to subsequent generations of cells. But failure to do this can lead to catastrophic cellular consequences. For example, the cell could die, or it could lead to the emergence of cells with altered properties that can result in disease, including cancers. When a cell divides, it duplicates all of its chromosomes and aligns them on what is known as a spindle structure. Microtubules, which are these long tubular-like arms emanating from the poles of this spindle, then grab onto these duplicated chromosomes and rearrange them around this spindle structure so that each side only has one copy of each chromosome. The cell then starts to pull apart, and the nuclear envelope or protective casing protecting the inner contents of the cell starts to reform around each of these pockets of duplicated chromosomes, and this then results in two identical daughter cells. My PhD focuses on this process and specifically on this alignment and segregation. Imagine if these chromosomes were not aligned correctly and instead were all over the spindle structure. Could the cell be sure that each chromosome had been duplicated successfully, or could it know that each cell was getting only one copy of each chromosome when trying to separate them out? This is why the alignment and subsequent segregation processes are vital for cells to divide correctly. Now, the microtubules don't work alone in governing this alignment process. They work alongside a complex of proteins known as the kinetochore, which sits on the chromosome. One of these proteins is called NDT80, which interacts with microtubules to aid this alignment and segregation. Now, as you can imagine, these proteins are very small and so can be quite tricky to visualize. So in order to combat this, I used CRISPR-Cas9 genome engineering technology. And what this effectively does is allows me to add in a tag known as a halo tag to the end of the protein, so that whenever the protein is made, the tag is also made. So if you think of the genome like one big long line of code comprised of just 4 base pairs, A, T, G, and C, in all different permutations and repeats. What I've effectively done here is found the code for the protein, and then just before it's about to end, I've added in the code for the tag. I then put this edited genome back inside of the cells by transfecting it, and then what will happen is whenever the cell makes the protein, it will now also make the tag. So whenever I add a fluorescent dye, it will bind to the tag and therefore allow me to visualize the protein. Now, what's great about using this halo tag is that it also has another function, as it allows me to bind what is known as a protag, which results in the protein being destroyed by the cell. Now, this destruction isn't instantaneous, which I can use to my advantage, as I could destroy, say, only 50% of the protein, and then see what effect this has on chromosomal alignment, and thus how vital it is for this process. I'm also looking at some of the enzymes that help to regulate this process, known as aurora kinases. I can again use CRISPR to add in fluorescent tags, or even knock out their functionality entirely, and check to see what effect this has on the system. By elucidating this system's behaviour during mitosis, I hope to understand how these aurora kinases regulate NDT80 to ensure accurate chromosome segregation and provide new insights into the way this altered regulation of the system drives genome instability in cancers.